This is the mini lesson on bloodstain science. The abbreviation BPA will stand for Bloodstain Pattern Analysis, and that's what these notes are going to be about, how we can look at stains left by blood at a crime scene to help us solve the crime. So the different things that you can learn by analyzing these blood stains is the type and velocity of the weapon. Remember, velocity would be how fast it's moving. You can determine the number of blows. So, say that someone was hit with a hammer, you can determine how many times. You can determine whether the person that was inflicting the injury, the assailant, if they were right or left handed. Positions and movements of the victim and the assailant during and after the attack. So, how they were positioned relative to each other throughout the crime. You could determine which wounds were inflicted first, the types of injuries, how long ago the crime was committed, and whether the death was immediate or if it took time, if it was a delayed death. So there's several different ways to detect blood at a crime scene. A light source can be used, so this will either be a high intensity light or UV light, and that will allow the investigators to find traces of blood at the crime scene that would not be visible under the normal lighting conditions from the lights that are in the home or from just regular light sources. You can also do blood reagent tests. So this means that a chemical is going to react with some component of the blood and cause a positive identification. These are referred to as presumptive tests. They're used to detect blood at crime scenes based on hemoglobin in the blood. So you would have to do more tests once you found the blood to determine whether it was human blood or not. But these tests can tell you whether there is any hemoglobin present in a substance if it's at a crime scene. So, some examples of these blood reagent tests: phenolphthalein is a chemical that is used. It's referred to as the Kastle-Meyer test, and it produces a pink color when it reacts with the hemoglobin in the blood. Hemastix is a strip that is coated with tetramethylbenzidine, TMB, and it produces a green or blue-green color in the presence of hemoglobin. Luminol, we've talked about before. So this is a chemical that investigators can use to look for traces of blood, even if it's been cleaned or removed with even bleach. So they'll put the luminol, spray it on where they think there might be blood, and they look for reactions with the iron that's present in the blood. And basically, you get this blue luminescence whenever you shine a particular light on it. Uh, Fluorescein is also a chemical that's capable of detecting latent or old blood by spraying it onto the substance or area that might contain blood. And you use a UV light or special goggles to detect any areas that would be illuminated. And these would appear as a greenish white color if blood was present. And then you have LCV, which stands for leucocrystal violet, and it's a type of chemical process that's used for blood enhancement. So this helps make the blood evidence more visible so it can be photographed and analyzed by the crime scene investigators. So when we talk about blood stain pattern analysis, you'll be using several new terms. Spatter. This is the blood stains created from the application of force to the area where the blood originated. Origin or source is going to be the place from where the blood spatter came from or originated. Angle of impact is the angle at which the blood droplet will strike the surface that it is left on. Parent drop, this is the droplet from which a satellite spatter originates. So you see in the image to the right, the parent drop is going to be the main component of the blood drop, and then spines can come off of that. Satellite spatters are small drops of blood that break apart from the parent spatter when the blood droplet hits a surface. And you can see that in the picture to the right as well. You get a little satellite spatter to the left of the parent drop. Spines are the pointed edges of a stain that will come out from the parent drop, kind of radiate out like rays of a sun, and they can help determine the direction from which the blood traveled. Types of bloodstain patterns, there's three main types. Passive bloodstains, these are patterns created from the force of gravity acting on the blood. So you'll get a drop, a series of drops, flow patterns, or when you get blood pooling into a big puddle. And that would be passive bloodstains. Projected blood stains are patterns that occur when a force is applied to the source of the blood. So think if someone was getting hit with something that caused them to bleed, that would be a projected blood stain. So this can include low, medium, or high impact spatters. They could be cast off, so like flung off of a weapon. It can come from arterial spurting, which would mean if you hit a main artery, there's a lot of force projecting the blood out. And then also expiratory blood blown out of the nose, mouth, or wound. The third type is a transfer or contact blood stain. This happens whenever um, a wet or bloody object comes in contact with another surface, and it can be used to identify an object or body part. And then a wipe is a pattern that's created from an object moving through a blood stain, while a swipe pattern is created from an object leaving a blood stain. So a wipe would be like if you walked through a puddle of blood and you created a white blood stain, whereas a swipe could be if you were bleeding on your hand and you moved your hand across the wall. Okay, so these are the notes for blood stain pattern analysis. Make sure they're filled out and glued into your notebook, and let me know so I can give you a stamp.